Hello, everyone. I'm Carter Clift, the CEO of Vascugen. We haven't presented at the MESA meeting before, so I'm going to use this time to give a general overview of the company and provide a little bit of specifics on where we're headed with our iPSC-based technology. The company is strictly an iPSC therapeutics company. It was founded roughly 2 years ago by myself and key opinion leader, Dr. Merv Yoder, based out of Indiana University. And we are building our pipeline and technology off an exclusive IP estate that was licensed from IU. Part of the strategy for our company is to develop everything in-house through early stage development, meaning that we have an R&D space that includes integrated clean room that allows us to do the manufacturing for initial IND enabling studies and clinical trials. And we thought that that was an important strategy for our company based on the experience that we'd had in the industry. And so the company is located in Madison, Wisconsin. We're in a very nice building, the former facility that was outfitted by Illumina. Myself, I come from the Fujifilm, CDI, and then subsequently the OPSIS therapeutics programs. I was involved in those and continue to be to some extent. Other members of our team come from companies like Mallinckrodt Stratotech, where they developed cell-based therapies that have now reached BLA. So they bring a lot of manufacturing expertise to our company, CMC, quality systems, GMP manufacturing, as well as folks from PPD in terms of the analytical side that provides some benefits for us. And then our CSO, co-founder, and essentially the science behind our technology is Dr. Yoder, who's widely published and has been pretty well established as one of the key figures in the endothelial lineage research out there. Our technology is really based on the notion of a human vascular stem cell, which naturally there would have to be such a cell in order to form all the blood vessels in the body during development and then to maintain them during homeostasis of the body, which obviously is a kind of flexible tissue in that it has to grow and be pruned over time depending on your physical state. And so what Dr. Yoder discovered in looking at the lineages of this vascular endothelium in much the same way that he and other groups have looked at the hematopoietic lineages in the past, he discovered that this cell, there is such a cell that's capable of forming de novo vasculature in vivo. It forms vessels in vitro like many endothelial cells, but it has a more robust function in vivo in that it can form durable vessels in ischemic animal models, for example. And so this is really the foundation for what we began developing from an academic setting 2 years ago into an industry setting today. One thing that I think is important to clarify about the technology and the approach is that it's fundamentally different from a mechanistic standpoint than most of the efforts that have been taken in the past to form new blood vessels, relieve ischemic conditions, increase blood flow to tissues where that's the problem in the disease. And that's typically done by angiogenesis, which is the process in which one attempts to cause sprouting of existing vessels. What we're really focused on is more of a cell replacement therapy. We're trying to replace in tissues that acutely need new vasculature. We're trying to reintroduce that cell, which is capable in and of itself of vasculogenesis. So it's in that sense not reliant on intrinsic vessel sprouting. It has a higher potential effect size if successful and safe. And I think it fits more with the vanguard of next-gen cell therapies that are being engineered from IPS technology. So about this cell, which is originally and currently in the literature called the endothelial colony-forming cell, or ECFC, it has a large number of different names that have been used for endothelial progenitors in the past, but this is the consensus that this is the vascular progenitor. And it naturally declines with age, as shown on the left from a study a number of years ago. But you could see there that there are a number of different metrics by which adult blood, AB, versus cord blood derived endothelial colony forming cells differ, whether it's the number of colonies that you can isolate from primary tissue, the percentage of cells that are dividing, the number of cells that you get per colony, all of these different metrics, you see a substantial difference. You also note that when measuring this population in normal versus disease, for example, type 2 diabetic patients, there is a significant difference in its potency. So there are clear kind of hallmarks that this cell type is affected by disease conditions and naturally during aging. Again, in the literature, historically, what people have looked at, for example, is infusing this into various models, isolating these cells and infusing them. And so here in a pig acute myocardial infarction model, there's an example of where infusion of the cells leads to substantially increased vessel formation in an infarct border zone. And that is, one of many different animal models. Another would be often people would be testing these in rodent hind limb ischemic models, in which you can show that you can rescue the limb function, you can restore blood flow to the limb, and that's exemplary on the right. In the ocular space, there is also data from multiple groups to suggest that for oxygen-induced retinopathy, in this particular case, you can relieve different aspects of the abnormal vascular area, whether it's completely avascular, whether it's neovascular, or if you're seeking ultimately to restore kind of normoxic conditions, is it normovascular? So this is another application where it may seem counterintuitive, but what one is attempting to do is to restore normal vascular function in the ischemic part of the retina that is in fact leading to dysfunction and aberrant vascular growth elsewhere. So because blood vessels are everywhere in the body, if one has the capability to introduce blood vessel forming cells therapeutically, it presents a large degree of optionality across many different tissues and disease states. And this list of PubMed accessions is just an example of the number of studies that are out there looking at different efficacy models. There are also 4 reviews that are on the bottom of our website if you wanted to learn more. So it becomes a little challenging to figure out exactly where to start once you have a cell that you're confident can form new blood vessels in vivo, that it's scalable, that it meets GMP compliance, for example. So this also brings up another important point regarding the cell, in that if you compare the ECFC as a potential therapeutic to other donor-derived cells, such as an MSC or a hematopoietic stem cell or immune cells like T cells, it's much less practical to develop a donor cell therapy from an ECFC just based on the difficulty in harvesting substantial quantities of it. So this really means that for this cell type to have meaningful application, it's going to have to be scaled most likely from an iPSC source, and that's what we're focused on. And that may explain in part why you haven't seen more of these donor-derived therapies of this nature entering the clinic. It's worth noting as well that the endothelial cells are varied just as any other complex lineage in the body, like a hematopoietic stem cell lineage, for example. And if you look at endothelial cells that are donor-derived versus ECFCs, you see a difference. In the case where Dr. Yoder and his group isolated iPS-derived iPS-derived ECFCs and compared them with off-the-shelf iPS-derived endothelial cells, there's substantial differences in the proliferative potential of the cells, and this is one of the hallmarks in which we're focused on as a functionality for the cells that we're developing. If one is trying to advance any cell therapy from an iPS technology, you end up, I guess, viewing it as a kind of engineering problem. The process development consists of an engineering problem. The delivery and therapeutic use of itself may become an engineering problem. And so we tend to look at this in that lens, in the sense of a development stack, basically, where we have a partner for iPSC-based technology, we have partners in process development, we have groups that we're working with, like Miltenyi, on advanced sorting technologies, such as their TITO system, for example. And then in terms of assay development, Nikon has a partnership with us in which we're looking at various imaging-based techniques to test and release and do basic R&D on our product. A really interesting one as well is the gene modification stack, if you will, in which we're working, with companies like Synthego and Aldevron, who are pioneering the ability to do this seamlessly in cells and then to transition it to GMP compliant manufacturing. And so we have identified a candidate and a few candidates. This particular iPSC-based product that we're scaling up now is one that compares quite similarly to an ECFC population that's of primary origin, in this case, cord blood-derived ECFC. So when looking at common endothelial antigens, there's a great degree of similarity and high purity across these antigens. And then across a suite of proprietary antigens that we're following and basing our development around, we see the same thing, essentially, that we see a very high expression of these antigens in our iPS-based product. Additionally, measures such as in vitro vessel formation, which may be a little bit challenging to see on the lower left to discern that, but suffice to say that we see very robust vessel formation in both of these cell types, and so that gives us confidence to move forward. Similarly, although we are yet embarking on a variety of different animal proof of concept studies, this one in particular with Maria Grant's lab at University of Alabama demonstrates that this target cell population can have a functional effect, as shown on the left based on an assay that they've developed to look at hypoxic signals. And so when in the lower, on the left side on the lower right, when delivering the product via intravitreal injection, it can relieve this ischemic condition as imaged this way. And on the right, we do see evidence of engraftment of these cells. And so we're looking into these things in more detail. From a pipeline standpoint, again, back to the point I made earlier about optionality, it can become a little bit difficult to determine exactly what the right target indications would be. And so we spent quite a bit of time on the diligence around that and continue to do so. What our ambition is and use of capital going forward is really to move 1 to 2 programs into IND based off of this candidate targeting diabetic retinopathy and then a peripheral ischemic condition. That's because those are areas where there is pre-existing proof of concept data and where we continue to generate that data. Beyond that, our ambition and what we're focused on with our current preclinical partners is to establish a kind of dashboard in which we can determine the application of the technology from a research standpoint in areas, for example, like GA or a couple of other undisclosed cardiovascular and we'll just broadly say tissue targeting approaches. And so like many IPS-based companies, we are early stage, aiming for IND, following in the footsteps of Fate Therapeutics or Cynata or Blue Rock, Sema, et cetera. So to sum up, again, just as an introduction to the company, we have what we think is a fairly unique iPSC-based vascular platform with novel IP and quite broad optionality. The mechanism in which the cells are thought to act and the basis for our advancing the programs is direct vessel formation or vasculogenesis. We decided as a company to build out both our team and our platform as an integrated fashion to support CMC, R&D development, and early stage manufacturing under one roof, basically, so that whomever we end up partnering with or as we advance, we will understand very clearly the quality attributes of our products. And just in terms of what we're looking for at this stage as a company, we'd be really interested to have pharma or device partners in vascular and or retinal. We think there's a lot of opportunity there, and we're a fairly nimble entity, so it would be pretty straightforward probably to proceed with such partners. And then at some point in the next year, we're planning to raise our Series A, and so we're also looking for a lead investor for that. Thanks for your time. If you want to reach out to me, feel free. My email is shown on this slide, and I enjoyed talking to you. Thanks.